This is Libby, and I'm going to be talking about examples of digestive enzymes. So I'm going to begin with amylase. Carbohydrates break down carbohydrates into sugars. Amylase is a type of carbohydrates that breaks down starch in our bodies. So the purpose of amylase is to break down starch into its constituent simple sugars, which is predominantly maltose. Amylase is produced in three places: the small intestine, the pancreas, and the salivary glands. And the sites of action of amylase are the small intestine and the mouth. So when amylase breaks down starch, maltose and other sugars are produced, as we've just learned. And the sites of action of amylase are the small intestine and the mouth, where it's used in digestion. Next, we're going to be learning about proteases. So proteases are digestive enzymes that break down proteins into amino acids. The sites of action of proteases are the small intestine and the stomach. The purpose of proteases is to break down proteins into amino acids. And proteases are produced in the small intestine, the pancreas, and the stomach. This differs to amylase, which is produced in the small intestine, the pancreas, and the salivary glands. So let's recap what we just learned about proteases. We know that they're produced in the small intestine, the pancreas, and the stomach. And proteases break down proteins into amino acids. The next digestive enzyme I'm going to talk about now is lipase. Lipase is a type of digestive enzyme that breaks down lipids into glycerol and fatty acids. Lipase breaks down lipids into a molecule called glycerol and fatty acids. Lipase is produced in the small intestine and the pancreas. So if we just recap these three digestive enzymes we've now learned about, we've got proteases, lipases, and amylase. Proteases break down proteins into amino acids, and they're produced and they function in the small intestine and the stomach. Lipase breaks down lipids into glycerol and fatty acids, and it is produced in the small intestine and the pancreas. Amylase breaks down starch into sugars like maltose, and it is produced in the small intestine and the mouth. So which enzyme breaks down lipids? We know this is lipase, and it breaks down lipids into glycerol and fatty acids. So how did amylase evolve? Humans have many more copies of the gene to make amylase than chimpanzees, our closest relatives. This is probably because our diet changed, so it's an advantage to break down starchy foods more easily. You don't necessarily need to learn this for your exam, but sometimes it's really helpful to have some context to understand why you're learning it. So proteases break down protein, which is in the name, into their constituent amino acids. So lipases are enzymes that break down lipids into two things. These are fatty acids and glycerol. And the action sites for amylase are, as we've learned already, the small intestine and the mouth. So your food starts to be broken down while it's still in your mouth before it reaches your stomach or your intestine. So let's think about the different functions of these three digestive enzymes. We know that lipase breaks down lipids into glycerol and fatty acids. We know that amylase, which is a type of carbohydrates, breaks down starch into simple sugars. And we know that protease breaks down proteins into their constituent amino acids. So that brings this session to a close. I hope that you feel like you've learned enough about these three examples. Thanks very much for listening.